telling us how c.elegans is used in experimental conditions. So, in this talk, I'm going to talk about how the literature, and there's a huge amount of literature generated on c.elegans, the huge knowledge base that has been generated can be used for theoretical simulations and modeling. So the question we have been asking is, do developmental constraints decide the structure of the c.elegans nervous system? So this work has been done in collaboration with Dr. Shitabra Sinha's group in Institute of Mathematical Science. So I'll be presenting a work which is done in collaboration. As we know that the structure of this c.elegans, the great thing is that it has only 302 neurons, out of which the number of neurons in the somatic nervous system is 279, and it's mapped out. So the connectivity among the neurons are known. We also heard that the whole within individual organisms, the position of the cells are invariant. So many different things are therefore can be mapped out and are already known. So in this work we have concentrated on the somatic nervous system. We have not considered the pharyngeal neurons. As you can see from the from this slide, have I got this? So the c.elegans has these clusters of neurons which are called ganglia. There isn't, of course, anything which is a central nervous system, but these ganglions have been defined as cells, as neurons which are in close physical distance, and in some ways the nomenclature and which neurons comprise which ganglia is arbitrary. The reason why we went into this work is based on some previous work that we have done in collaboration. So in that PLOS paper, what we had done is we have used modularity. We have looked at modularity in the neuronal network structure of c.elegans, and we have used the structural analysis to identify critically important neurons. As you can see from the left hand side panel, we found that there are some neurons which fall under what we call local hubs, some which form under connector hubs, and some which have been identified as part of global hubs. So by hubs we mean, let us suppose that an individual member of a family is very important as part of for the family fabric. So this individual talks to all members of the family, interacts with them, and therefore as far as the family is concerned, as far as the local hub is concerned, this is a very important member of that hub. On the other hand, there may be somebody in the same family who remains aloof as far as family is concerned, but has lots of friends with outside people. So those would be therefore connector hubs. And there may be the other extreme where you have a family member who not only interacts with other members of the family but also with outside contact. So these are the people who would be classified as being global hubs. So what we had shown from this work is that the structural analysis can actually help us identify components which are really critically important. One of the predictions that we have made is that AVKL and SNBVL are important neurons. Again, this is undetermined as far as experiments are concerned, but from our work we think that given their position, these are neurons which are likely to be of importance. So given that from structural analysis of the neuronal network we could identify these functional important neurons, it suggests that function defines structure to a certain extent, but of course that's not the only thing which is defining structure, and development is one of the more important aspects which also define the structure. So we went on to look at how developmental constraints are going to decide the structure of the C. elegans nervous system, and we find some interesting patterns emerging. So again, the development of C. elegans has been mapped out extremely well. They come from a single cell zygote, and of course have the same genetic composition. And as Sulston's work showed, almost every cell has been mapped out, and along with the mapping of which cell derives from which cell, it's also known which of these cells are neurons and which of these neurons connect to each other. As far as neurons are concerned, they mostly derive from AB cells. So from single zygote, C. elegans divides into AB and P1, and most neurons derive from AB. One of the first things we looked at is, if you look at birth time, then what is the percentage of the different neuronal types which appear at the different stages of the organism. So C. elegans neurons are divided into, as we have already heard, sensory neurons, interneurons, and motor neurons, depending grossly on their function. And we have divided in this image why am I not being able to? Great, thanks. So we have divided into embryo, L1 stage, L2 stage, and L3 stage, and as you can see here, and this is certainly intuitive, sensory neurons and interneurons, 90% of them appear in the embryonic stage before 500 minutes, while motor neurons, to reach that same amount of same fraction, that takes place only at the L2 stage. This is not unexpected, but this basically shows us that we can actually validate some of the things that are intuitively we know. So C. elegans neurons, of course, have both chemical synapses and gap junctions, and both in terms of connectivity matrix is again completely mapped out, whether which neurons have synapses with which neurons and which neurons share gap junctions with which neurons. So using that connectivity matrix, we have divided the neurons. And as you can see in this slide, we have a matrix. So we have divided it into short projection length neurons, medium projection length neurons, long projection length. This is the presynaptic neuronal process length we are considering. Now, by short, we usually mean 0.3 fraction of the body length. Medium is 0.5, and long is greater than 0.6 fraction of the body length. So these are the neurons with presynaptic connections of these lengths. These are the postsynaptic connections that are received, and then we have divided each of these squares into the birth time. So this is the lowest birth time, this is the highest birth time. Further, it has been divided into before hatching versus after hatching, and each of these cells basically represents a neuron cell distance, the physical distance between the cell bodies. Whenever the neuron has a connection partner, synaptic connection partner, it's labeled in different color, and depending on how close the cell bodies are. And when they don't share a connection, it's basically colorless, uncolored. So from this slide, we realize that typically short neurons possessing short presynaptic process length versus short postsynaptic process lengths are quite dense in number, and as you can see here that most of the cells which have hatched before tend to form processes among themselves. So this is true for short versus short, then medium versus short again, and in this instance also. There are a couple of exceptions, and those exceptions actually suggest a few things. For example, this is a neuron with a long presynaptic process forming connections with a neuron who has a short process length, but after hatching period. So similarly, this is again another instance of a case which is similar to this, in which instance this is again after hatching. The postsynaptic, the long neuron with postsynaptic connections form a connection with a set of neurons with long presynaptic connections, but again after hatching. So these kind of exceptions suggest that physical distance probably is not the only thing, and it's likely that there is an axon guidance mechanism which allows say presynaptic neurons with long processes to hunt up postsynaptic neurons with short processes. So along with this is a derivation of the previous slide. So what we have basically done is those neuronal pairs which have connections between them. If you look at this average cell distance bodies, we have just averaged it out in this instance. As you can see, white means that the cell distance bodies are very close together. The darker it is, it means they're further apart. So if you look at these portions, so higher average distance between cell bodies seem to occur between long presynaptic neurons connecting with short postsynaptic neurons in comparison to, say, when long postsynaptic neurons connect to short presynaptic neurons. So this is also true for, in fact, medium neurons connecting with short process postsynaptic neurons. So, so this gives an idea of what is the average cell distance between the different classes of these neuronal divisions. So, along with the synaptic, the neurons forming a synaptic connectivity matrix, we also did the gap junctional connection distance, and more or less they have a similar profile. Of course, it's important to note that along this diagonal, there's a prominence of connections which typically represent the symmetrically paired generated at a lineage distance within 15 to 20. About 17 is the peak. So we went on to look at how synaptic connections occur as a function of physical distance, lineage distance, and average birth time of connected neurons. So basically, in this case, we have this gray plane shows the hatching time, and if you look at the different axes, first you have the time versus the lineage distance, and the most important points in this is the bimodality that occurs at low lineage distances. You have a lot of neurons growing at this time and a lot being generated at here, but this becomes homogeneous when lineage distance increases. Again, if you look at average birth time and physical distance, what we find is that at higher average birth times, the probability of connections between neurons with cell bodies which are far apart is reduced, as you can see from the number of neurons here. And finally, we find that very few synapses are generated between neurons which have low lineage distance, if you look at this, and this is regardless of whether these neurons occurred before hatching or after hatching. So again, we have looked at the gap junctions as a function of physical distance and lineage distance, and pretty much the trend is similar. One important thing to note is that there seems to be fewer occurrences of gap junctions at higher average birth time. So that's again a little bit intuitive, so it validates some of the things that we know about c. elegans. So next thing we looked at is what kind of constraints are imposed by lineage distance on the development of ganglia. We started with ganglia. So what we have done here is we have calculated the mean inter and intraganglionic lineage distance, and if you look along this diagonal, you can see the lineage distance, the mean of the lineage distance is very low. It's all below mostly 15. So, so that basically suggests that neurons are more closely related to neurons of the same ganglia rather than neurons of other ganglia. When we did the coefficient of variation of the same ganglia, however, we came upon a rather striking result. In fact, it's contrasting, I would say, because there's a relative variability in lineage distance along the neurons of a ganglion is larger than between neurons belonging to different ganglia. Again, you can see that these are in fact a lot higher than the surrounding instances. So I just want to point out that the neurons have been arranged from this end. So we have numbered the neurons numbered from this end. So this is how the ganglions are arranged. So the next thing we looked at is constraints imposed by lineage distance when it comes to connection probability, and the most noticeable thing is that the connection probability of synapses falls as lineage distance increases. In fact, the p-value is extremely low with a very very good correlation coefficient. This correlation is much weaker for gap junctions. As you can see, there's hardly a trend there, and that's borne out also by the p-values. When we look at symmetric paired neurons versus ones which are unpaired, we come up with some striking results. We find that the number of synapses forming between symmetrically paired neurons, the probability of connection is much higher as compared to the ones which are unpaired. And this holds true for bidirectional synapses and for gap junctions also, which you can see is really shows a high probability of connection. So we went on to look at the joint probability density for given lineage distance and time difference. So in this instance, we looked at all neurons, not just connected pairs of neurons. As you can see, there's a increasing probability density in birth times between two neurons as the lineage distance increases, and there's a very interesting trend seen at very low birth time differences here, where there's an alternating rise and dip in the probability density as we go along the lineage distance. Therefore, the probability of low birth time difference is high for odd lineage distances and low for even lineage distance. We'll explain that in a minute. So this is how we measured the lineage distance, as we had discussed, that it's going to be, even if you have a lineage distance from A to B is 3, 1, 2, 3, and what you notice is that neurons, that for even lineage distances, neurons can never be of the same rung, whereas for odd lineage distances, neurons can be of the same rung. So hence the probability of low birth time differences are high at odd lineage distance and low at even lineage distances. it's important to note that neurons at the same rung are more or less born close in time. So we went on to look at, if you look at number of cell divisions and birth time, pretty much up to the 10th cell division, there's most of the motor, inter and sensory neurons show a very large number of cells being generated, and there is a big jump in the rung after the 10th division. As you can see, there's a huge number of cells being generated in L1, L2 after the 10th cell division. So if you look at the, this is not very visible from this particular graph, but if one looks at the cumulative numbers, in fact, there is a dominance of sensory neurons in the lower rung of lower rungs, while motor neurons are generated more in the higher rungs of the graph. So motor neurons basically are generated later again, and sensory neurons are generated earlier on. So having gone through all this, we of course wanted to look at whether the functional circuit neurons, which have been all experimentally identified, can show up some pattern with an analysis like this. So we have considered 8 functional circuits, that's individual egg laying, thermotaxis, chemosensory, defecation, locomotion, and locomotion has been divided into 3 circuits: satiated while feeding, hungry, which is exploration, and tap withdrawal. So I think the audience by now is primed about the tap withdrawal circuit, which has been so well presented. So what we went on to look is how the development of the functional circuits, how does that fit in with time of formation, and we find that majority of functional circuit neurons are born early. As you can see, most of them are born while the worm is still in the embryonic stage. So this is again expected, because functional circuit neurons by their definition are generated because of their because they are of such fundamental importance, and expectedly feeding, exploration, chemosensation, thermotaxis are really important for the worm's survival, and therefore are almost fully complete in many instances before the organism is hatched out. And if you look at egg-laying circuit, which is of course a function of the adult organism only, it's one of the slowest developing circuits, as you can see here. It develops only at the fag end of L1 stage. So, that's expected. That's expected in the biological sense. It really makes sense. The next thing we looked at is the degree and projection length. So by degree we mean the number of connections received and sent out by the neuron. And again, what we find is that if you look at all neurons, the out degree, which is the number of connections going out of the neuron, versus the in degree, is pretty much same. But this is not true for functional circuit neurons. We find that if you look at the average degree, the out degree is higher than in degree. And again, within functional circuit neurons, there seems to be some fine tuning happening within early functional circuit neurons and late functional circuit neurons. So late functional circuit neurons show more out degree processes, while early functional circuit neurons show lower number of out degree processes. This has been dissected out much more in these 4 panels. Again, we have dissected out in degree and out degree with early neurons and late neurons. Particularly noticeable is that in the late neurons, this tail end with is missing. So basically, we are showing here that what's the probability of early neurons having a larger number of degree, and we find that the early neurons have a very high probability of having a large number of connections. This could be very much because since they have been born early, they tend to form more number of processes, whether they receive processes or whether they have given out processes. That's not happening in neurons which come late in the after hatching, for example. So that's one thing which comes out when we look at the probability of occurrence of synaptic connection. The other thing that we looked at is the fraction of neurons that's with different projection lengths. Again, we have divided it into short, medium, and long, and we have looked at which functional circuit has which type of neurons, whether they have neurons with medium length processes more or whether they have neurons with long length processes more. And when we do that, we find that tap withdrawal and mechanosensory circuits have neurons which are predominantly long and medium processed neurons. This is the one, if you just average out, so you can see what's the huge differences there is. In fact, if you average out, you can see that most neurons are ones with short processes, with medium and long process length neurons being almost equal in fraction. While in mechanosensation circuit, you can see that in fact the number of neurons with short processes is not there at all. Most neurons have long processes and some have medium processes. So, that's actually a surprising result of. But given that mechanosensation and tap withdrawal required the whole organism's participation in the movement, it's probably not so surprising. So, therefore, functional circuit neurons tend to have a higher degree than average, and late neurons have much lower maximum degree than early neurons. So, to summarize what this analysis showed, that even by looking at the developmental constraints, we can tease out circumstances which probably define why the c. elegans nervous system structure developed as it is. So, neuronal length of neuronal projections and their birth times are very important for governing the connectivity of the whole somatic neuronal network. And spatial distance between cell bodies do not limit formation of synapses. This suggests that there is a guiding mechanism for neurons with long projections forming connections with short projection neurons. Lineage distance between two neurons is a measure of genetic relatedness, no surprises there. And it affects several attributes of c. elegans neural networks, such as connection probability, spatial distance between neurons, and difference in birth times. So, all these things are in fact decided by the lineage distance between two neurons. The spatial organization of neurons in ganglia is affected again by the lineage history, since neurons in the same ganglia are more closely related lineage-wise than neurons belonging to different ganglia. And as we saw, functional circuit neurons, by definition being some of the more important neurons, develop quite early. Most of them develop very, very early. So, they also have a very high degree in comparison to average, the average neuron, and neurons generated at later birth times show limited connectivity compared to early neurons. So, I want to thank Anand Pathak, who did under the supervision of Dr. Shitabra Sinha for this work. And we have, with the support of Department of Biotechnology, began some work with the experimental work with c. elegans being done by Pavitra, my student. Thank you.
Questions? question. It's here. You showed us how the synapses are forming, but with the growth, do you also see deletion of synapses, or is the circuitry so precise that every synapse is?
So, in C. elegans, the way these synapses have been found out is at certain stages, people have done hundreds of electron micrographs. So, I don't know of electron micrographs being able to show deletion of synapses. So, these are very much ones which have stayed throughout the stages that people have looked at those electron micrographs. So, at whatever stages electron micrographs have been generated, those synapses have been, but it doesn't show any erasing of synapses at all.
Another related question is that is there anything in terms of development of correlation between degrees, in and out degrees?
Yes, so in fact, there is a correlation of in and out degrees. So, development, we already showed that birth time in fact decides the number of degree, whether it's in or
no, I
out.
mean the number of degree, but I meant the target. So, pre and post synaptic targets, is there a change across development stages? So, let's say if I take two neurons, if they have common presynaptic nodes or common postsynaptic nodes, and if that evolves with time somehow.
I don't think I got your question. I mean, early neurons tend to form connections, as we saw with neurons born later also, which is not necessarily true. So, neurons coming up after hatching form much less connections with neurons which have hatched before. So, but the number of connections is probably a fact of a neuron being around for a longer time period. So, early neurons in fact have larger number of connections in and out degree combined. Evidence to see whether, okay, I would really like to talk to you about it. No, we have not looked at it. I mean, I would really like to know which strains, etc., to look at. Because, most of it, some of it is intuitive, what we find. But experimental validation of some of the findings we have seen, some of the patterns we have seen would be wonderful.
So if we
can I ask one more? Yeah. So just a short one. So what fraction of these total connections they end up in functional circuits as opposed to non-functional?
I think that data should be out somewhere. So basically what you're asking is how many synapses and gap junctions do functional circuit neurons produce as opposed to no. I don't think that's particularly striking, if I remember rightly, that they have a larger number of connections with compared. So as we saw, the number of in degree and out degree actually is variable in functional circuit neurons as compared to an average. You're suggesting that they hog all the synapses. I don't think that's necessarily true. I mean, C. elegans people maybe, I don't think there is that out of the 6,000 synapses, most of them belong to functional circuit neurons. It probably doesn't work. Yeah.
So if there are no other questions, we are almost back on time. And Shita. Yeah, so we should thank Nivedita for her presentation.